Welcome back to the deep dive. We're the show that, takes these really complex scientific topics and tries to give you that definitive shortcut to getting your head around them. For, generations, medical diagnosis was, macroscopic, wasn't it? Looking at cells under a microscope, seeing organ damage, watching symptoms develop. But today, that world has just been completely flipped on its head. We're not just looking at the downstream effects of disease anymore. We're actually reading the body's instruction manual, the DNA itself. So our mission in this deep dive is a pretty rigorous, but I think essential exploration. We're looking into the molecular analysis of genomic alterations. And just to be clear, we are focusing squarely on the techniques, the lab methods we use to spot those tiny but incredibly critical changes in DNA and RNA, the changes that cause disease, define it, and increasingly tell us how to treat it.
Absolutely. And if you're trying to get a handle on these methods, the very first question, the most fundamental one is, where is this genetic change actually located? It sounds simple, but this distinction drives everything that follows: what sample you collect, which technique you choose. And we basically split them into two main buckets.
Okay.
First, you've got constitutional markers. Now, think of these as being part of the blueprint, present in literally every single cell right from conception. A classic example. Maybe the CFTR gene mutation that causes cystic fibrosis, or the gene alteration in Huntington disease. It's everywhere. So because it's universal in that person, you can usually get your sample easily. Peripheral blood is common, or even just a cheek swab. You extract the DNA, and you're good to go.
Right. That makes intuitive sense. The problem is systemic, so the test can be too.
Right.
But the second category, somatic markers, that sounds like where things get more complicated, especially with something like cancer.
It's exactly right. Somatic markers are, well, they're localized. They're restricted to specific tissues or lesions, or even just certain cells within a lesion. Think about RAS mutations in colorectal cancer, or those fusion genes you find in leukemias. Those changes are only in the cancer cells. They're not in the patient's healthy blood cells or skin cells.
So the sample has to be the tumor itself.
Precisely. You need that biopsy tissue or the surgical resection specimen. Or if you're looking at an infectious disease, say tuberculosis, the mycobacterial DNA you're hunting for is going to be concentrated at the site of infection, maybe the lung tissue. So this difference, constitutional versus somatic, it's the absolute first branching point for any molecular diagnostic workflow. You have to match the test strategy to where that alteration actually lives.
Okay, so sample choice is clearly paramount. And, before we dive into the specific techniques we use today, it's probably worth taking just a moment to sort of zoom out and appreciate the history here. This field moved incredibly slowly for a long time.
Oh, absolutely. Think about conventional karyotyping, literally looking at stained chromosomes under a microscope to spot something like Down syndrome, or older methods like southern blotting for Huntington's. These were incredibly manual, slow, low-throughput processes. You were maybe looking at one or two things at a time, max.
Really painstaking work.
Very. Then came, I guess, the breakthrough decade or maybe decade and a half. 1977, we get Sanger sequencing, the first practical way to actually read a DNA sequence.
Right.
Then, just 6 years later, 1983, Kary Mullis comes up with the polymerase chain reaction, PCR.
The molecular photocopier.
Exactly. And those two inventions together just turbocharged molecular biology. Suddenly we could amplify tiny bits of DNA and read their sequence relatively quickly.
But even that was just setting the stage, wasn't it? The real revolution, the thing that's put us on this steep curve we're on now, that came later, right? Late 90s, early 2000s.
That was the arrival of massively parallel sequencing, what we now just call next-generation sequencing, or NGS.
And that shift is still echoing through, everything in healthcare.
It absolutely is. We are squarely in a transition period. You see NGS systematically starting to replace many of those older, simpler single-gene tests. And the reason is just economics and speed.
It's that cost-performance curve.
It's staggering. I mean, you can sequence an entire human genome now for, under $1,000?
Yeah.
And get the data back in days. Compare that to the cost and time even a decade ago, let alone 30 years ago.
It's night and day.
Which is why nucleic acid testing has moved from being this niche research thing to being absolutely central to clinical practice.
And the need is just enormous. You mentioned two areas earlier where molecular diagnostics are really sort of leading the charge: cancer and infectious diseases.
Definitely. In oncology, these tests aren't just confirming a diagnosis anymore. They're dictating the entire treatment plan. We find specific tumor alterations, think HER2 amplification breast cancer determining Herceptin use, or EGFR mutations in lung cancer pointing towards specific targeted inhibitors.
So it guides therapy choice directly.
Directly. And we also use it to monitor treatment. We look for minimal residual disease, that tiny fraction of cancer cells that might survive initial therapy. Using techniques like quantitative PCR for the BCR-ABL fusion transcript in chronic myeloid leukemia, or CML, lets us see if the treatment is truly working at a molecular level.
And cancer isn't static, is it? Tumors evolve, especially under the pressure of treatment.
That's a critical point. If a patient responds well initially to a targeted drug, but then the cancer comes back, the molecular lab goes hunting again. We look for new secondary mutations that have emerged in the tumor genome, mutations that confer resistance to that drug.
It's like an evolutionary arms race inside the patient.
It really is. And in infectious disease, it's similarly crucial. Molecular tests give us definitive diagnosis. We detect the pathogen-specific DNA or RNA. We can also spot genetic changes in the microbe that make it resistant to certain antibiotics or antivirals.
Right, like MRSA or drug-resistant TB.
Exactly. And critically, we monitor treatment effectiveness by quantifying the amount of the pathogen, think viral load measurements, for HIV or hepatitis C. It tells you if the drugs are suppressing the infection.
So the stakes are incredibly high. We need technologies that are not just accurate, but also exquisitely sensitive. Okay, let's start unpacking the toolbox. First set of techniques. Focusing on detecting those relatively small changes, point mutations, maybe small insertions or deletions, the kind of thing a standard PCR approach can usually handle. And you mentioned it already, PCR, the polymerase chain reaction. It really is the workhorse, isn't it?
It is foundational. And the concept is, well, elegantly simple when you think about it. You take a tiny amount of DNA, maybe even just one starting molecule, containing the specific short segment you care about, usually less than, say, 1,000 base pairs. Then you use a special heat-stable DNA polymerase enzyme, primers that flank your target region, and cycles of heating and cooling in a thermal cycler. Each cycle doubles the amount of your target DNA fragment.
So exponential amplification.
Exponential. You go from one copy to billions in just a couple of hours.
Yeah.
It generates enough material that you can then actually analyze it properly.
Okay, so you've amplified your target sequence using PCR. Now you need to read it or analyze it somehow.
Yeah.
For a long time, the gold standard for actually determining the exact sequence was Sanger sequencing, right?
Correct. Sanger sequencing was the dominant method for decades, and it's still used. It's a very clever piece of biochemistry. Yeah. You take your amplified PCR product, and you run a new reaction. You include primers, the polymerase, normal DNA building blocks, the nucleotides A, T, C, and G. But you also add a small amount of special terminator nucleotides.
The dideoxy ones.
Exactly, dideoxy terminators. There's one for each base, A, T, C, G. And critically, each one is labeled with a different fluorescent color.
Okay.
Now, when the polymerase is copying the DNA strand, it mostly incorporates the normal nucleotides.
Yep.
But occasionally, randomly, it incorporates one of these fluorescent dideoxy terminators. And when it does that, the chain can't be extended any further. It stops.
A dead end.
A dead end. So what you end up with is a whole collection of DNA fragments, all starting at the same primer, but ending at every possible base position along the sequence, each tagged with the color corresponding to that final base.
I see.
Then you separate these fragments by size, using capillary electrophoresis, basically, pushing them through a very thin tube filled with the gel matrix. The smallest fragments zip through fastest, the largest slowest.
And a laser reads the color as they pass.
Precisely. A laser excites the fluorescent tags as the fragments pass a detector, and the order of the colors tells you the DNA sequence base by base.
It sounds very elegant, and it's known for being accurate. But you hinted at a limitation earlier, especially for cancer work. What's the catch with Sanger?
The catch is heterogeneity. Sanger sequencing works beautifully when your starting sample is pure, like testing for an inherited constitutional mutation where every cell has the same sequence. But think about a tumor biopsy. It's almost never pure tumor cells. It's a mix. Tumor cells, normal stromal cells, infiltrating immune cells, blood vessels.
Oh, right, a real mix.
So if you sequence a PCR product from that mixed sample using Sanger, what you get is essentially an average signal at each base position. If a specific mutation is only present in, say, 30% of the cells in that sample, the signal from the mutant base is often too weak to be clearly distinguished from the strong signal coming from the normal base present in the other 70% of cells.
So low-frequency mutations get missed.
They get masked by the background. Sanger isn't sensitive enough to reliably pick up mutations below roughly 15–20% frequency in a mixed sample. which is a major problem in oncology, where clinically important mutations might be present at much lower levels, especially early on or in minimal residual disease.
That's a critical limitation. So that's where next-generation sequencing, NGS, comes in. Even when you're just sequencing a relatively short PCR product, it overcomes this sensitivity issue.
Fundamentally, yes. The core difference is massive parallelism. Instead of sequencing one reaction in one capillary tube, NGS platforms sequence millions or even billions of individual DNA molecules simultaneously in separate little reactions all across a chip or flow cell.
So you're not getting an average signal, you're getting millions of individual reads.
Exactly. This gives you what we call sequencing depth.
Yeah.
You might sequence the exact same base position hundreds, thousands, or even tens of thousands of times across all those parallel reads.
Okay.
So even if a mutation is only present in, say, 5% of the original DNA molecules in your sample, it will still show up in roughly 5% of your sequencing reads. And because you have so many reads overall, that 5% signal is statistically robust and easily distinguishable from random sequencing errors.
The depth gives you the statistical power.
Precisely. For cancer diagnostics, where you're almost always dealing with these mixed cell populations, that ability of NGS to reliably detect low-frequency variants is absolutely crucial for finding actionable targets or tracking resistance. It's a game changer in terms of sensitivity.
Okay, so NGS offers that deep dive into the sequence. But sometimes you don't need the whole sequence of the PCR product, right? Sometimes you know exactly which base you need to check for a specific known mutation. Are there faster, maybe cheaper PCR-based methods for those kinds of spot checks?
Yes, absolutely. If you know the precise location and the specific base change you're looking for, there are highly targeted and often very sensitive PCR-based approaches.
Like what?
Well, one very common and sensitive technique is single-base primer extension. This is frequently used for mutations that are very common hotspots, like the BRAF V600E mutation you see in melanoma and other cancers.
How does that work?
You design a PCR primer that anneals to the DNA template, immediately adjacent to the specific nucleotide position you're interested in, literally one base upstream.
Okay, right next door.
Exactly. Then you perform a reaction with just polymerase and special terminator nucleotides again, fluorescently labeled. But this time, the reaction conditions are set up so that the polymerase only adds one single base to the end of your primer. Just one. You might include, say, a green-labeled terminator for the normal base and a red-labeled terminator for the expected mutant base at that position. The polymerase adds the complementary base, and because it's a terminator, the reaction stops. You then measure the amount of green versus red fluorescence. The ratio tells you precisely the proportion of normal versus mutant DNA at that single target nucleotide. It's very fast, relatively inexpensive, and can be exquisitely sensitive for detecting known point mutations. But of course, it only tells you about that one specific base.
Highly focused, then. What about something a bit simpler conceptually? I remember hearing about using restriction enzymes, like molecular scissors.
Yes, restriction fragment length analysis, or RFLP. That's an older but sometimes still useful technique. The idea relies on restriction enzymes.
Right, those enzymes that cut DNA only at very specific recognition sequences.
Exactly. Each restriction enzyme recognizes a specific short DNA sequence, maybe 4 to 8 base pairs long, and cuts the DNA whenever it encounters that sequence. So if the specific point mutation you're interested in happens to either create a new restriction enzyme recognition site or destroy an existing one, you can use this difference.
How?
You amplify the region containing the mutation site using PCR. Then you digest that PCR product with the relevant restriction enzyme. If the mutation is absent, normal DNA, the enzyme will cut or not cut in a specific way, producing DNA fragments of predictable sizes. If the mutation is present, the cutting pattern will be different, producing fragments of different sizes.
And you see that difference on a gel.
Exactly. You run the digested products on an electrophoresis gel. The normal and mutant alleles will produce different banding patterns because the fragment sizes are different.
Simple enough, but it sounds like it depends entirely on whether the mutation happens to fall within a convenient restriction site.
That's the major limitation. It only works if the mutation perfectly aligns with a known restriction site, and if the mutation at that site is always the same base change, it lacks flexibility. But when it works, it can be a very straightforward and inexpensive test for a known invariant mutation.
Okay. And PCR is also really good at detecting changes in the length of a DNA segment, right? Not just point mutations, but insertions or deletions.
Yes, absolutely. That's what we call amplicon length analysis. This is critical for diagnosing a whole class of disorders caused by unstable expansions of repetitive DNA sequences, particularly trinucleotide repeat disorders.
Like Fragile X syndrome.
Fragile X is the classic example. It's caused by an expansion of a CGG repeat sequence in the promoter region of the FMR1 gene on the X chromosome.
Okay.
In the normal population, people might have, say, 5 to 44 CGG repeats. Individuals with a premutation might have 55 to 200 repeats, and individuals with the full mutation causing Fragile X syndrome have over 200 repeats, sometimes thousands.
So huge differences in length.
Huge differences. So we design PCR primers that sit outside this entire repeat region. They flank it. When we run the PCR, the length of the amplified product, the amplicon, directly reflects the number of repeats present.
And you just measure the size.
You measure the size, typically by running the PCR products on a gel or using capillary electrophoresis. You can easily distinguish the small normal alleles from the larger premutation alleles and the much, much larger full mutation alleles based purely on their size. It's also used for other repeat expansion disorders like Huntington's disease or myotonic dystrophy.
But is there a limit? Can PCR handle really massive expansions?
That's a crucial technical point. There is a limit. Conventional DNA polymerases used in PCR can struggle with very long, highly repetitive sequences. They can sort of slip during replication, leading to inaccurate amplification or even complete failure to amplify the largest alleles.
So if someone has thousands of repeats in Fragile X,
If the expansion is exceptionally large, standard PCR might fail to amplify the full mutation allele altogether, or it might produce a smear instead of a distinct band. In those cases, PCR becomes unreliable or uninformative for the full mutation.
what do you do there?
You have to fall back on older, more robust, but also more cumbersome techniques, specifically southern blotting. This involves cutting the patient's genomic DNA with restriction enzymes, separating the large fragments by gel electrophoresis, transferring them to a membrane, and then using a labeled DNA probe that binds specifically to the FMR1 region to visualize the fragments containing the repeat
So you measure the size directly from the genomic DNA.
directly. Southern blotting can accurately size even those massive expansions that choke standard PCR. It's a great example of how sometimes the older technology remains essential as a backup or for specific challenging situations. It's not always about the newest thing being the only answer.
That's a really interesting point, technology supplementing, not just replacing.
Okay, before we leave PCR, we have to touch on its quantitative power. Measuring how much of something is there, not just if it's there. Real-time PCR.
Yes, real-time PCR, also often called quantitative PCR or qPCR, this is a hugely important technology in the clinical lab. Unlike traditional PCR, where you just look at the end product, qPCR monitors the amplification process as it happens, cycle by cycle.
How did it do that?
It uses fluorescent reporters. These can be dyes that bind to double-stranded DNA, like SYBR green, or specific fluorescent probes, like Tachman probes, that only fluoresce when they bind to the target sequence during amplification.
So the more product you make, the brighter the signal gets.
Exactly. The instrument measures the fluorescence intensity in real time during the exponential phase of the PCR reaction. The cycle number at which the fluorescent signal crosses a set threshold is inversely proportional to the starting amount of target DNA.
Meaning, the more target you started with, the fewer cycles it takes to see the signal pop up. Precisely. By comparing the sample's threshold cycle to a standard curve generated from known quantities, you can accurately calculate the starting concentration of your target sequence in the original sample.
So you get a number, not just a yes-no. What are the key applications?
They are fundamental for clinical monitoring. We use it extensively in oncology, for instance, to quantify the level of specific cancer markers in blood or bone marrow. Tracking the BCR-ABL fusion transcript levels in CML patients on therapy is a prime example. It tells the clinician exactly how well the targeted drug is suppressing the leukemia cells.
Monitoring minimal residual disease.
Exactly. And it's also standard practice for monitoring infectious diseases. Measuring viral load, the number of HIV RNA copies per milliliter of blood, for instance, is crucial for managing HIV patients and assessing antiviral therapy effectiveness. Same for hepatitis C, or monitoring viruses like Epstein-Barr virus or CMV in transplant recipients who are at risk of reactivation. qPCR gives you that critical quantitative measure of disease burden or treatment response.
Okay, that really covers the small-scale analysis. Beautifully, point mutations, small repeats, quantification using PCR. But, as you said, sometimes the genetic problem isn't small. What happens when we're dealing with alterations on a much grander scale? An entire chunk of a chromosome missing, or a huge piece duplicated, or complex rearrangements, things measured in kilobases or megabases, way too big for standard PCR.
Right, that's the next major conceptual pivot. When you move from gene-level alterations to chromosome-level or large genomic region alterations, you need techniques that can survey much larger territories. And the core principle behind most of these large-scale methods is hybridization.
Hybridization using a labeled piece of DNA or RNA, a probe, to find its complementary sequence within the patient's genome.
Exactly. You're using a known sequence to find its match within the vast complexity of the cell's DNA. And the classic visually based hybridization technique for large-scale changes is fluorescence in situ hybridization, or
FH. I always picture those brightly colored chromosomes.
That's essentially what it is. With the H, we use relatively large DNA probes. These can range from tens to hundreds of kilobases in length. These probes are chemically labeled with fluorescent dyes.
Different colors for different probes?
Often, yes. You can use multiple probes with different colors simultaneously. You then take the patient's cells, these could be dividing cells, showing condensed metaphase chromosomes, or non-dividing cells with interphase nuclei, prepare them on a microscope slide, and apply these fluorescent probes. The probes then diffuse into the cells and bind, or hybridize, specifically to their complementary target sequence on the chromosomes within those cells.
And then you look under a fluorescent microscope.
You got it. Where the probe is bound, you see a bright fluorescent signal pinpointing the specific chromosomal region. It's literally like using a microscopic fluorescent highlighter to paint the exact stretch of DNA you're interested in.
It sounds incredibly powerful for visualizing specific large targets. What are the main reasons you'd reach for FIH in the clinical lab?
Its main strength is detecting large-scale genomic abnormalities that might be too subtle or complex for standard chromosome banding analysis, karyotyping, to resolve clearly. This includes things like confirming numeric chromosome abnormalities, aneuploidy, like an extra chromosome 21 in Down syndrome, detecting smaller microdeletions or microduplications that involve deleting or adding just a small piece of a chromosome, often associated with specific genetic syndromes, identifying complex chromosomal translocations, where pieces of two different chromosomes have broken off and swapped places. SH probes flanking the breakpoint can show abnormal fusion signals, and crucially, assessing gene amplification.
Amplification making many extra copies of a specific gene.
Yes, this is critical in cancer. For example, FAAH is considered the gold standard method for determining the copy number of the HER2 gene in breast cancer cells. If the HER2 gene is amplified, meaning there are many extra copies lighting up with the FAH probe, the patient is likely to benefit from targeted therapy like Herceptin trastuzumab.
So it directly guides treatment.
Directly. Similarly, measuring amplification of the NMYC gene by FISH in neuroblastoma is a key prognostic factor. And another important aspect is speed. FIH on interphase nuclei can often provide results faster than waiting for cells to divide for traditional karyotyping. This is vital in urgent situations, like suspected acute promyocytic leukemia, APL. APLs is caused by a specific translocation involving the retinoic acid receptor gene, and treatment with all trans-retinoic acid needs to start immediately. FIH can rapidly confirm that translocation, allowing treatment to begin without delay.
Okay, speed and targeted visualization are clear advantages, but there's a limitation, isn't there? With FIH, you have to know what you're looking for, right? You need to choose the right probe for the suspected abnormality.
That is the fundamental limitation of FIH. It's a targeted approach. You have to have specific hypotheses about which chromosomal region or gene is altered. If the critical genetic lesion is somewhere unexpected, or if it's a type of abnormality you didn't anticipate, FAHH won't find it because you weren't using the right probe.
So if you don't have a specific suspect, FISH isn't the right tool. You need something that casts a wider net.
Exactly. If you need a global survey of the entire genome for copy number changes, without necessarily knowing where to look first, you need to move beyond FISH. And that brings us to cytogenomic array technology, essentially using microarrays to scan the whole genome at once.
Right, microarray platforms, giving us that genome-wide perspective. The first major type was array-based comparative genomic hybridization, or array CGH. How does that work?
Array CGH is a clever comparative technique. You take the patient's DNA, the test DNA, and label it with one fluorescent dye, say cyanine 5, which fluoresces red. Then you take a normal reference DNA sample from a healthy individual and label it with a different dye, say cyanine 3, which fluoresces green.
Okay, red for patient, green for normal.
Right. Then you mix these two labeled DNA samples together in equal amounts and apply them to a microarray chip. This chip is covered with thousands or even millions of tiny spots. Each spot contains a short, known DNA sequence, a probe representing a specific location across the entire genome.
So the patient's DNA and the reference DNA compete to bind at each spot.
Precisely. They compete for hybridization to the probes on the array. After hybridization, you use a scanner to measure the intensity of the red and green fluorescence at every single spot on the array.
And the color tells you the copy number.
It does. If the patient has the normal 2 copies of the DNA sequence represented by a particular spot, their red-labeled DNA and the green-labeled reference DNA will bind in roughly equal amounts. The resulting mixture of red and green fluorescence looks yellow to the scanner.
Yellow means normal.
Yellow means normal, diploid. Now, if the patient has a deletion in that region, they only have 1 copy, or 0 copies, compared to the reference's 2 copies. So less red DNA binds, the green reference DNA dominates, and the spot appears green.
Green means deletion.
And conversely, if the patient has a duplication or amplification in that region, 3, 4, or more copies, their red DNA outcompetes the green reference DNA. More red binds, and the spot appears red.
Red means gain.
Exactly. By analyzing the red-green fluorescence ratio across all the spots spanning the entire genome, the computer can generate a high-resolution map showing precisely where the patient has gains, duplications, or losses, deletions, of DNA compared to the normal reference. It was a massive leap forward in resolution compared to traditional karyotyping for detecting these copy number variations, or CNVs.
Okay, array CGH gives us a really detailed count of DNA segments across the genome. But then the technology evolved again, didn't it? We got single nucleotide polymorphism, SNP, genotyping arrays. How are these different, and what extra information do they provide?
SNP arrays represent the next level, and they've largely replaced array CGH in many clinical settings. They leverage a different feature of the genome, single nucleotide polymorphisms, or SNPs.
SNP is those single base pair differences between individuals.
Yes. SNPs are the most common type of genetic variation in humans, occurring roughly every 1,000 base pairs or so. They act like millions of tiny landmarks scattered throughout the genome. Now, SNP arrays work slightly differently than CGH. Instead of just measuring total signal intensity, they are designed to determine which specific allele, which version of the SNP, the A version or the G version, is present at each SNP location across the genome.
So it's not just how much DNA is there, but which version of the DNA sequence is there at these specific SNP landmarks.
Exactly. SNP arrays can certainly detect copy number variations, CNVs, just like array CGH, often by looking at the overall signal intensity at each SNP. But the crucial added dimension is the genotype information, or zygosity data. It tells you whether at a given SNP locus, the patient is homozygous, for example, AA or GG, meaning they inherited the same allele from both parents, or heterozygous, or it's ADAG, meaning they inherited different alleles from each parent.
Okay, so it gives you copy number and genotype information. Why is that extra genotype zygosity information so clinically powerful? What does it let you see that CGH fundamentally misses?
This is where SNP arrays deliver those really crucial diagnostic insights. They are, of course, excellent for finding CNVs, deletions, and duplications, often in children with developmental delay or birth defects, whereas standard karyotype look normal. That's a common application.
But the real power is beyond just CNVs.
The real power lies in their ability to detect loss of heterozygosity, or LOH. This is something array CGH is completely blind to.
Okay, explain LOH and why it matters.
LOH means that at a particular region of a chromosome where an individual should be heterozygous, having inherited different alleles from each parent, like AG, they are instead homozygous, like AA or BB. They have lost the heterozygosity they were expected to have.
But the total amount of DNA might still be normal.
That's the key. You can have LOH even when the copy number is perfectly normal, deployed. Array CGH would just look at the total signal, see it's normal, and report no CNV detected. It completely misses the underlying allelic imbalance.
So where is this clinically critical?
The classic example is diagnosing disorders caused by uniparental disomy, or UPD. This is where an individual inherits both copies of a particular chromosome, or part of a chromosome, from the same parent, instead of 1 copy from each.
Like Prader-Willi and Angelman syndromes.
Exactly. These syndromes can be caused by UPD involving chromosome 15. If a child inherits both chromosome 15s from their mother, maternal UPD, and none from their father, they have the normal number of chromosome 15s too, so array CGH would look normal. But because both copies came from the mother, the child will be homozygous, show LOH, across large stretches of chromosome 15 at SNPs where the mother herself is homozygous.
Ah, the SNP array sees that lack of paternal contribution.
It sees the abnormal stretch of homozygosity instantly. It detects that allelic imbalance even though the copy number count is normal. This ability to detect LOH is absolutely essential for diagnosing UPD-related disorders, which CGH simply cannot do. It's like checking the parental origin stance on the chromosomes, not just counting them.
That's a fantastic illustration of the added diagnostic power. And does this ability to track alleles also help with detecting mosaicism?
Yes, it can. Mosaicism, where an individual has two or more genetically distinct cell populations, can sometimes be detected by SNP arrays. If a significant proportion of cells have a CNV or LOH, it creates characteristic shifts or skewing in the allele frequency plots, the B-allele frequency plots, generated by the SNP array analysis. Experienced cytogeneticists can often recognize these complex patterns as suggestive of mosaicism, sometimes even when the abnormal cell line is present at relatively low levels. So SNP arrays give you count, parental origin via LOH, and clues about mosaicism a much richer picture than copy number alone.
Okay, that wraps up the large-scale genomic analysis really well. So let's pivot now to section 3. Beyond the basic DNA sequence, we need to talk about situations where maybe we can't test the gene directly, or where looking at RNA is actually more informative, or even where the crucial changes aren't in the sequence at all, but are chemical modifications on the DNA.
Right. Let's start with using polymorphic markers and linkage analysis. This is more of a diagnostic strategy than a single technique, and it's particularly useful in specific circumstances.
When would you use linkage analysis?
You typically turn to it when the actual disease-causing gene for a known inherited disorder is maybe still unknown, or perhaps it's very large and technically difficult or expensive to sequence directly in every family member you need to test.
So you use a workaround.
Essentially, yes. The principle is genetic linkage. The idea is that two genetic loci, positions on a chromosome that are located very close together, are physically linked and therefore tend to be inherited together through generations. They rarely get separated by recombination during meiosis.
They travel together.
They travel together.
Yeah.
So if you can't directly test the disease gene itself, you find a nearby genetic marker, a polymorphic site, whose inheritance pattern you can easily track within the family.
And you see if that marker travels with the disease.
Exactly. You genotype all the available family members for this marker locus. If you consistently see that a specific version allele of the marker is always inherited by the family members who have the disease, and rarely or never by those who don't, you infer that the disease gene must be located very close to that marker on the chromosome. You're using the marker as a surrogate to track the inheritance of the unseen disease allele.
What kind of markers are typically used for this?
Historically, repeat-length polymorphisms were heavily used, and still are for certain applications. These are regions of DNA containing short, repeated sequences. There are two main types. Mini-satellites, or variable number tandem repeats, VNTRs, have repeat units typically 10-100 base pairs long. Microsatellites, also called short tandem repeats, STRs, have much shorter repeat units, usually just 2 to 6 base pairs long, like CA repeated over and over.
And the number of repeats varies a lot between people.
Hugely. That's why they're so useful. These repeat regions are highly polymorphic. Different individuals have different numbers of repeats, meaning the length of that DNA segment varies greatly in the population. This high variability makes them incredibly informative for distinguishing between individuals and tracking inheritance within families. SNPs are also used now for linkage, especially in genome-wide studies, but STRs remain important.
So, beyond tracking disease genes indirectly, what are the main applications of analyzing these polymorphic markers like STRs?
Their high variability makes them ideal for applications requiring genetic identity. This includes forensic medicine, comparing STR profiles from crime scene samples to suspects, paternity testing, comparing a child's STR alleles to the potential fathers, transplantation, confirming identity and relatedness between donors and recipients, and a really important one in hematology oncology, monitoring chimerism after stem cell or bone marrow transplantation.
Chimerism, the mix of donor and recipient cells.
Exactly. After transplant, you want the donor cells to take over the patient's blood system. But using PCR to amplify informative STR markers that differ between the donor and the recipient, you can quantify the percentage of donor cells versus recipient cells in the patient's blood over time.
So you can see if the graft is taking.
You can see if the graft is engrafting successfully increasing percentage of donor cells. And, importantly, you can detect early signs of graft failure or disease relapse if the recipient's original cell markers start to reappear or increase. It's a critical monitoring tool post-transplant.
Okay, that makes sense. Now let's switch gears to RNA analysis. You mentioned earlier that DNA is generally preferred in the lab because it's much more stable. RNA is notoriously fragile, so when is it absolutely necessary, or maybe just much better, to analyze RNA instead of DNA?
That's a great question because you're right, RNA's instability does add technical challenges. But there are several key situations where RNA analysis is essential. First, obviously, if you're trying to detect or quantify RNA viruses, things like HIV, hepatitis C, influenza, their primary genetic material is RNA, so you have to target RNA. Second, in cancer research and sometimes diagnostics, analyzing mRNA expression levels can be very informative. Looking at which genes are highly expressed, transcribed into mRNA, versus lowly expressed can help classify tumors into different molecular subtypes that might have different prognosis or respond differently to treatment. This is often done using techniques like RNA sequencing, RNA-seq, or microarrays.
So profiling gene activity.
Right. But perhaps the most critical diagnostic reason for choosing RNA analysis, especially in cancer, relates back to detecting certain chromosomal translocations that result in fusion genes.
Like the BCR-ABL fusion in CML we talked about earlier.
That's the canonical example. Remember how we said translocations often happen within large introns, the non-coding regions of genes?
Yeah, deep inside the gene structure.
Well, trying to design PCR primers to amplify across that huge, often poorly defined intronic breakpoint region in the genomic DNA can be extremely difficult, if not impossible. The distance is too large, the sequences might be repetitive or unknown.
But RNA processing solves that problem.
Exactly. During RNA processing, those large introns are spliced out to create the mature messenger RNA, mRNA. So if the translocation brought exon 1 of the BCR gene next to exon 2 of the ABL gene, the resulting fusion mRNA transcript will have BCR exon 1 directly joined to ABL exon 2, a much shorter, well-defined sequence.
Ah, splicing conveniently removes the problematic introns.
Precisely. So the standard technique is to first extract the RNA from the patient's cells, then use an enzyme called reverse transcriptase to convert the RNA back into a stable DNA copy called complementary DNA or cDNA.
RNA to cDNA.
Right. Now that cDNA copy of the BCR-ABL fusion transcript is relatively short and lacks the introns. It's a perfect template for PCR amplification. You can easily design primers in the BCR exon and the ABL exon to amplify across the fusion junction in the cDNA.
Much easier than trying to amplify the genomic DNA breakpoint.
Much, much easier and far more reliable. This technique, reverse transcription, followed by PCR, RT-PCR,
Right.
especially real-time quantitative RT-PCR, RT-qPCR, is the absolute standard of care for detecting the BCR-ABL fusion transcript and for monitoring minimal residual disease levels in CML patients. It offers incredible sensitivity because you're targeting the abundant mRNA transcript.
So for certain fusion genes, RNA analysis via cDNA is the way to go. Okay. Finally, in this section, let's talk about epigenetic alterations. This is the layer of control above the DNA sequence itself, right? Modifications that change how genes are used without changing the A's, T's, C's, and G's.
Exactly. Epigenetics refers to heritable changes in gene function that occur without altering the underlying DNA sequence. These are often chemical modifications either directly to the DNA bases or to the histone proteins that package the DNA into chromatin.
And these modifications are important.
They are absolutely critical for normal development and cellular function. Think about processes like genomic imprinting, where only the maternal or paternal copy of gene is expressed, or X chromosome inactivation, where most genes on one X chromosome are silenced in females. These rely heavily on epigenetic mechanisms.
And they're also involved in disease, particularly cancer.
Hugely involved in cancer. One of the most well-studied epigenetic modifications is DNA methylation. This typically involves adding a methyl group, CH3, to cytosine bases, specifically where they occur next to a guanine base, the so-called CpG dinucleotide.
CpG sites.
Right. And regions rich in these CpG sites, called CpG islands, are often found in the promoter regions of genes, the sequences that control gene activation. Generally, heavy methylation of a CpG island in a gene's promoter region is associated with silencing or shutting off the expression of that gene.
So methylation acts like an off switch.
Often, yes. And abnormal methylation patterns silencing tumor suppressor genes or activated OnQ genes are hallmarks of many cancers.
Okay, so if we want to diagnose conditions related to abnormal methylation, we need a way to detect this chemical change, since the underlying DNA sequence, C versus C methylated, looks the same to a standard sequencing reaction. How do we do that?
We need a chemical trick to distinguish methylated cytosine from unmethylated cytosine. The workhorse method for this is treatment with sodium bisulfite.
Sodium bisulfite, what does it do?
It's quite clever. Sodium bisulfite treatment chemically modifies DNA in a way that specifically converts unmethylated cytosine bases into uracil bases, which are then read as thymine, T by DNA polymerase during subsequent PCR sequencing.
Okay, unmethylated C becomes U, read as T.
But, and this is the crucial part, methylated cytosines are resistant to this chemical conversion. They are protected by the methyl group and remain as cytosines.
So the treatment creates a constituents difference.
Exactly. After bisulfite treatment, you perform PCR and sequence the region of interest. Now, any position that was originally an unmethylated C will appear as a T in your sequence read. Any position that was originally a methylated C will still appear as a C.
So you can directly read the methylation pattern from the sequence.
You can map methylation status base by base. This bisulfite sequencing approach, or variations like methylation-specific PCR that use primers designed to recognize bisulfite-converted sequences, is essential for diagnosing disorders where abnormal methylation is the primary cause.
Like which ones?
We mentioned Fragile X syndrome earlier, caused by the CGG repeat expansion. That expansion leads to hypermethylation and silencing of the FMR1 promoter. Bisulfite analysis is used to confirm this silencing methylation pattern. It's also crucial for those imprinting disorders like Prader-Willi and Angelman syndromes, which are often caused by deletions or UPD, but can also result from primary defects in the methylation imprint itself on chromosome 15. You need methylation analysis to diagnose those cases.
Fascinating. So chemical treatment unlocks the epigenetic information. Okay, we've journeyed through small changes, large changes, linkage, RNA, and now epigenetics. Let's bring it all together and return to the technology that's really reshaping the entire field, the one that holds the promise of potentially unifying many of these approaches. Next-generation sequencing, NGS. This is where things get really interesting, isn't it?
It really is the driving force behind so much progress right now. As we touched on, NGS fundamentally changed the game because, unlike Sanger sequencing, which needed that clean, single template, NGS thrives on complexity. It's designed to handle massive amounts of DNA, often from highly heterogeneous samples like tumors or microbiomes.
And the power comes from doing millions or billions of reactions at once.
That massive parallelism is the key. And while different NGS platforms use slightly different chemistries, Illumina's sequencing by synthesis is the dominant one. But there's also Ion Torrent's semiconductor sequencing, and others they generally share three core principles.
Okay, what are they?
First, spatial separation. You have to somehow isolate individual DNA fragments from each other physically so you can sequence them independently. On Illumina platforms, this is often done by attaching fragmented DNA molecules randomly to a dense lawn of primers coating the inside surface of a glass flow cell.
So each fragment gets its own little patch of real estate.
Kind of, yeah. Second, local amplification. A single DNA molecule usually doesn't generate enough signal to be detected reliably during sequencing. So you need to amplify each isolated fragment in situ right there on the flow cell surface. This is often done by a process called bridge amplification, where the fragment bends over and primes off a nearby primer, creating a copy, and you repeat this cycle a limited number of times.
Creating a small cluster of identical copies.
Exactly. You end up with millions of these spatially separated clusters, each derived from a single original DNA fragment. Each cluster now contains enough identical molecules to generate a detectable signal.
And the third principle is the sequencing itself.
Right. Parallel sequencing. All these millions of clusters on the flow cell are sequenced simultaneously. In the sequencing by synthesis approach, fluorescently labeled nucleotides and polymerase are washed across the flow cell. As the polymerase adds the complementary base to the growing strand in each cluster, the incorporated nucleotide emits its characteristic color of light, which is captured by a camera.
One base at a time, across millions of clusters at once.
One base at a time, cycle after cycle. The instrument records the sequence of color changes for each cluster. This generates millions or billions of relatively short sequence reads, typically maybe 50 to 300 base pairs long, depending on the run setup. It's the sheer volume of these reads that gives NGS its power, speed, and cost-effectiveness per base compared to Sanger.
Incredible scale. So how is this power actually deployed in the clinical lab? It sounds like you could do anything from focusing on a few genes to doing the whole genome.
And that's exactly right. The application really depends on the clinical question and balancing factors like cost, turnaround time, and the complexity of data analysis. We generally think about three main scales of clinical NGS application.
Okay, what's the most common?
By far the most common clinical use today is targeted sequencing, often using pre-designed gene panels.
So focusing the sequencing power on a specific list of relevant genes.
Precisely. You might have a cardiomyopathy panel with, say, 50-100 genes known to cause inherited heart muscle diseases, or a solid tumor panel covering maybe 500 genes frequently mutated in various cancers, including known drug targets and prognostic markers.
Why use a panel instead of sequencing everything?
Several reasons. First, by focusing only on the genes of interest, you can achieve very high sequencing depth for those genes reading each base hundreds or thousands of times. This maximizes your accuracy and sensitivity for detecting variants, especially low-frequency ones in tumors. Second, it significantly reduces the amount of data generated compared to whole exome or genome, which makes the analysis and interpretation much faster and more manageable. Third, it's generally the most cost-effective NGS approach right now.
So targeted panels are used for diagnosing inherited disorders with known gene associations and for detailed tumor profiling to guide cancer therapy.
Exactly. And also for things like repeat testing on a tumor. When a patient develops resistance to a drug, the panel might include genes known to acquire resistance mutations.
Makes sense. Okay, what's the next step up in scale?
The next step up is whole exome sequencing, or WES.
The exome, that's all the protein coding parts of the genes, right?
Correct. The exome represents only about 1.5% to 2% of the entire human genome, but it's where the vast majority, 85% of known disease-causing mutations are located. So WESS aims to sequence just these protein coding regions, the exons, of nearly all 20,000 human genes.
Still targeted, but much broader than a panel.
Much broader. WES has been incredibly successful, particularly in pediatric genetics, for finding diagnoses in children with complex, often mysterious syndromes or developmental disorders, where previous testing, including targeted panels, failed to find an answer. It casts a much wider net for finding those rare, unexpected genetic causes. It's often called the workhorse for diagnosing orphan diseases or ending long diagnostic odysseys.
Okay, so WES covers the coding regions. What's left? The ultimate analysis.
That would be whole genome sequencing, or WGS, sequencing essentially everything, all 3 billion base pairs, including the exons, introns, the vast intergenic regions, regulatory sequences, everything.
That sounds like the most complete picture. Why isn't WGS the standard first-line test then?
Primarily, it comes down to cost and complexity, though costs are dropping rapidly. WGS generates a truly enormous amount of data, making the storage, analysis, and particularly the interpretation incredibly challenging. You find millions of variants compared to the reference genome, and figuring out which one, or ones, might be clinically significant, especially those in non-coding regions where our understanding is still limited, is a major hurdle.
The variance of uncertain significance problem gets magnified.
Massively magnified. However, WGS does offer diagnostic advantages that WES fundamentally misses. While WES is great for finding mutations within genes, it's not very good at detecting structural variants, where the breakpoints lie outside the coding exons, deep in introns or in those intergenic regions.
What kind of structural variants?
Things like large deletions or duplications where the edges are in non-coding DNA, complex rearrangements, or translocations where the breakpoints might be far away from any actual gene coding sequence, perhaps involving regulatory elements. WGS, because it sequences everything without the targeted capture step used in WES, provides a much more comprehensive view of the genome structure.
So WGS is better for finding novel structural rearrangements relevant to certain cancers or developmental disorders.
Yes. If WES fails to find an answer, especially if a structural variant is suspected, WGS becomes the next logical step. It's currently the only sequencing method that can provide that truly comprehensive structural view alongside the sequence information.
Okay, so we have panels, exomes, and genomes. But generating the sequence data, even billions of bases, is only half the battle, isn't it? You mentioned the interpretation challenge. This brings us squarely to the crucial role of bioinformatics.
Absolutely essential. You cannot do clinical NGS without a robust bioinformatics pipeline and skilled bioinformaticians and clinical scientists to interpret the results. As you said, a single NGS run can generate hundreds of billions of base pairs of raw sequence data. Turning that data deluge into clinically meaningful information is a massive computational and intellectual task.
Can you walk us through the basic steps in that pipeline? What happens after the sequencer spits out the raw reads?
Sure, it generally involves several key stages. First is alignment or mapping. The software takes those millions or billions of short sequence reads and tries to figure out where each one belongs on the reference human genome sequence. It's like assembling a giant jigsaw puzzle where you have billions of tiny pieces and a picture of the final puzzle on the box lid, the reference genome.
Okay, lining up the reads against the map.
Exactly. Once the reads are aligned, the next step is variant calling. This is where the software systematically compares the patient's aligned sequence reads to the reference genome sequence at every single position. It looks for differences, single nucleotide changes, SNPs, small insertions, or deletions in guidals.
And the sequencing depth helps here.
Critically, if you have 100 reads covering a specific base and 99 of them show a G while the reference has an A, you can be pretty confident that the patient has a genuine G variant there. If only 2 out of 100 reads showed a G, you'd be much more suspicious. It might just be a sequencing error. Depth equals confidence in variant calling.
Makes sense. So you get a list of variants. What next?
This is arguably the most critical and complex step. Variant annotation and interpretation. Just having a list of differences isn't useful. You need context. The pipeline automatically annotates each variant with information like, which gene is it in? Does it change the protein sequence, the dissonance-nonsense frame shift? How common is this variant in population databases? Have clinical databases like ClinVar reported this variant before? And if so, was it classified as pathogenic, benign, or a VUS, a variant of uncertain significance? Does prediction software think this amino acid change will damage the protein?
Pulling in data from lots of sources.
Massive amounts of data. And then, skilled clinical scientists and geneticists review the annotated variants, especially the rare or potentially damaging ones, in the context of the patient's clinical presentation, their phenotype. They make the final judgment call on which variants are likely responsible for the patient's condition and need to be reported back to the clinician.
And that report needs to be clear for the doctor.
Absolutely. A crucial part of the process is writing a clear, concise report that explains the findings, classifies the variants according to established guidelines, like the ACMG guidelines, and details the clinical significance and implications for the patient and potentially their family. The interpretation is key. Generating the data is becoming easier. Interpreting it wisely is the real challenge.
It really sounds like the analysis is just as important, if not more so, than the sequencing itself. And I've heard about an even more advanced layer of analysis emerging, looking for patterns, mutational signature calling.
Yes, that's a fascinating and rapidly evolving area, particularly in cancer genomics. The idea is that the total pattern of mutations found in a tumor's genome can sometimes reveal clues about the underlying processes that cause those mutations. It's like genomic forensics.
How does that work?
Different mutational processes like damage from UV light, exposure to tobacco smoke carcinogens, or defects in specific DNA repair pathways tend to leave characteristic fingerprints or signatures in the genome. These signatures might be specific types of base substitutions, example, C to T changes are common after UV damage, or particular patterns of small insertions and deletions. Bioinformatic algorithms have been developed that can analyze the complete set of mutations detected by WGS or even WES in a tumor and deconvolute them, identifying the contribution of known mutational signatures.
So you can infer the history of the tumor's development.
To some extent, yes. And sometimes this has direct clinical relevance. The prime example right now is detecting a signature indicative of microsatellite instability, SI, or mismatch repair deficiency.
MSI high tumors.
Exactly. Tumors that are MSI high have a defect in their DNA mismatch repair system, leading to thousands of mutations, particularly small insertions and deletions in repetitive microsatellite regions. It turns out that these MSI-high tumors, regardless of their tissue of origin, are often highly responsive to treatment with immune checkpoint inhibitor drugs immunotherapy.
So identifying that signature predicts treatment response.
It's a powerful predictive biomarker. Clinical labs now routinely use NGS data and these signature algorithms to determine the MSI status of tumors, which directly informs whether immunotherapy is a good option for that patient. It's a great example of moving beyond just finding single mutations to interpreting the overall mutational landscape.
That's really powerful. So looking ahead, where is all this heading? It feels like NGS is poised to take over even more territory.
I think that's undeniable. The trajectory is clear. Costs are continuing to fall, speed is increasing, and the technology is improving. NGS is likely to supplant many, if not most, of the individual genetic tests we've discussed today.
Even things like RNA analysis.
Yes, RNA sequencing, RNA-seq, is becoming increasingly common. It allows you to not only detect fusion transcripts like BCR-ABL, but also to quantify the expression levels of all genes simultaneously, providing a much richer picture than single-gene qPCR. As costs drop, comprehensive RNA-seq might become standard alongside WGS or WES.
What about emerging applications?
There are several exciting areas. Microbiome analysis using NGS to characterize the vast communities of bacteria, viruses, and fungi living in and on us is a huge research area with potential clinical implications. Liquid biopsy is another major focus.
Testing blood for cancer DNA.
Exactly. Detecting tiny amounts of circulating tumor DNA, ctDNA, shed from tumors into the bloodstream. The hope is to use NGS on ctDNA for early cancer detection, monitoring treatment response without needing repeat tissue biopsies, and detecting minimal residual disease or emerging resistance mutations. The sensitivity required is immense, but the progress is rapid.
And the sequencing technology itself is still evolving.
Oh, yes. We're already seeing the clinical adoption of what are sometimes called third-generation sequencing technologies. These platforms, like those from Oxford Nanopore or PacBio, offer long-read sequencing. They can sequence single DNA molecules, often tens or even hundreds of thousands of base pairs long, without the need for PCR amplification first.
What's the advantage of long reads?
They're much better at resolving complex structural variants, mapping repetitive regions of the genome that are difficult for short reads, and potentially phasing alleles, determining which variants are on which chromosome copy, directly. As these technologies mature and their accuracy improves, they will likely play an increasingly important role, perhaps complementing or eventually even replacing short-read NGS for certain applications. The field is constantly moving.
It's an incredible pace. Okay, let's try to synthesize all of this. What's the big picture takeaway for someone trying to navigate this complex landscape?
We've covered a huge amount of ground, haven't we? From using restriction enzymes to check a single base pair, to Fairy-H lighting up whole chromosome regions, SNP arrays mapping copy number and LOH genome-wide, and NGS providing sequence data at scales from targeted panels to the entire genome.
A vast toolkit.
A vast and powerful toolkit. But I think the single most important lesson is that no single technique is universally optimal. The choice of diagnostic test absolutely requires deep knowledge of the specific type of genetic alteration you're looking for, the clinical context, and the limitations of each method.
You need to match the tool to the job.
Precisely. Think back to the cystic fibrosis example. A standard mutation panel might work brilliantly for someone of Ashkenazi Jewish descent because it covers their common founder mutations. But that same panel might have very poor sensitivity for someone of Asian descent who likely has a different spectrum of CFTR mutations. You need close communication between the clinician ordering the test and the laboratory performing and interpreting it to ensure the right test is chosen for the right patient and the right question.
Context is everything.
And that leads directly to the second major takeaway, which we've touched on several times, the interpretation challenge. As the technology, especially NGS, allows us to generate ever more comprehensive genetic data, the bottleneck is increasingly shifting from data generation to data interpretation. The bioinformatics analysis, the variant classification, understanding the clinical significance, that's where the complexity and, frankly, the current limitations often lie.
The wisdom is harder to generate than the data.
Well put. We're moving from just identifying mutations to interpreting complex genomic landscapes and mutational signatures. The expertise required to do that accurately and responsibly is paramount.
Which brings us to our final provocative thought for you, the listener.
Considering the relentless drop in cost and increase in speed for whole genome and whole transcriptome RNA-seq sequencing, if we reach a point where getting a comprehensive WGS and RNA-seq analysis is as fast and cheap as, say, a standard blood count today, will that kind of global, comprehensive molecular analysis become the default first diagnostic test for almost any condition?
Yeah.
Will it simply replace the entire constellation of specialized targeted tests? The single gene assays, the FIV-ASH probes, the specific panels that we've spent this whole deep dive discussing?
A single test to rule them all.
Perhaps. But if that happens, imagine the consequences. Every patient entering the healthcare system potentially having their entire genome and transcriptome sequenced up front. The sheer volume of data would be astronomical. The number of variants of uncertain significance uncovered would explode.
Who interprets all that?
Exactly. Who interprets it? How do we manage incidental findings? How does our healthcare system and how do individual clinicians and patients cope with that level of complex, often ambiguous predictive information? The technological capability might soon be there, but are we as a system, as professionals, as individuals, ready for the interpretive and ethical tsunami that follows? That interpretive bottleneck might be the biggest challenge facing the future of molecular medicine. Something to think about.